Now, genomics is kind of very interesting because in the past decade or so, it became almost free for you to sequence your genome. It's kind of like a few $100s. If you catch a good deal, then probably maybe around $200. So anybody can realistically have their genome sequenced. However, if you want to actually interpret your genome, that gets much, much more interesting because none of the labs really give you proper analysis. So they can give you a nice report which says, well, specifically for me, they gave the report that I like strength sports more than athletic sports. I'm like, okay, amazing, thank you, but that doesn't really help me in my life. Alternatively, you can tell them that, hey, I want a full report of everything, and they will give you back a 10,000 item list with various findings and literally hundreds or thousands of papers referencing them, and good luck, have fun. So it's like there's no in-between. And then I figured that, well, you know what, guys, I'm just going to analyze my own genome. I mean, how hard could it be? Well, that turned out to be a less enlightened idea. Turns out that in the past 80 or so years, there's a lot of papers have been written on genetics and on DNA. So just in the last year, I think we're up to about 54,000 papers. And if you think that the breadth is the only problem, actually depth is the other crazy problem. I highlighted a paper written by one of my friends. I have no idea what it means, and life is too short for me to understand what that paper actually means. This guy was doing eye research for 15 years. And there's no way I'm going to understand his research results that he spent 15 years figuring out. And this is a very, very interesting problem in this world of genomics because you have the results, you have the data, you have the papers, you have everything, except a meaningful way to apply that knowledge to yourself. And then, of course, obviously, probably for everyone in the room, the first tool that nowadays you would reach to is, well, let's try to use some AI tools. Let's try to use some LLM, because the LLMs are super good exactly at interpreting that data. So if you can get them to interpret that data and apply it for us, you know, problem solved. Everything, we're on the happy path. And it actually kind of works. So a few weeks ago, I had a presentation at ETH Prague, and somebody on the hallway challenged me to write an app, kind of like a toy app for a hackathon, that can tell you how well you tolerate alcohol, or how well you like alcohol. You know, a completely scientific test for alcohol tolerance. And I figured that, okay, that's fun. We can do it in a couple of hours. So I just shot at Claude that, okay, Claude, write me an app that can analyze my genome and tell me how well I tolerate alcohol. And the first thing Claude does is just scours the internet, scours PubMed, downloads a lot of articles, and identifies a few very, very interesting points of genetic interest. For example, when you drink alcohol, what happens behind the scenes is that alcohol gets transformed into acetaldehyde. I'm not sure that's the correct pronunciation. That's the thing that your body really, really hates. And afterwards, that thing gets digested, metabolized, and you're happy. And interestingly, this pathway, this converting alcohol into this middle gunk and then digesting it, has a few genes influence that. One of the genes kind of influences how fast you can digest that gunk. Most people digest it at some baseline speed, and if you have one single mutation, it means that you digest it about 15 times slower. So that alcohol, that gunk, remains in your body for 15 times longer. Another gene, for example, defines how fast you produce this gunk. And typically, people in Africa and Asia, they have this, I wouldn't call it flaw, this quirk in their genome, which means that they produce this gunk really, really fast, like 100 times faster than people in Europe or America. And these are really, really cool things that Claude just found out. Or Claude also went in and found some other really interesting points of interest, like how whenever you drink alcohol, it produces endorphin, it produces dopamine, it produces various other stuff. And now, depending on how your body reacts to these, you can be happy, you can be sad, a lot of things can happen. And when I saw this report, I mean, Claude probably produced it in like 5 minutes, max, maybe less. I'm like, hey, holy shit, this is working. This is the future. And I ran it against my genome, and that's when reality kicked in. Claude made a few mistakes, to say the least. For example, your DNA is kind of like 2 strands, and these 2 strands are complementary each other. It's like if one strand has a number 1 in it, the other strand has a number -1 in it, something like that, very, very hand-wavy. But the research literature has these things mapped out for one of the strands, and when you sequence your genome in the lab, you get the other strand, and Claude never made the link that, hey, you need to transform one into the other. So basically, it operated on completely junk data. Or the other thing, Claude pulled out that, hey, I'm interested in this genomic position, and when copy-pasting the position from the research paper into its code, it went 25 kilobase pairs off. Why? I don't know. Some AI hallucination. And interestingly, or for example, a few other issues that happened is that whenever you have your genome sequenced, you don't get it back that this is your genome. Rather, what they do is they give you a diff that compared to the reference human, these are the changes. And the reason they do that is because a lab works with a lot of genomes, so for them it's much easier to store 1,000 diffs and 1 reference versus to store everybody's genome raw. But then Claude just looked at it that, hey, this thing that I have is, what was the issue exactly? Oh, yes. So I have it the same way as the reference genome has, so all good. Yeah, but the reference genome has it wrong. I mean, the average human has that flaw. And then Claude, instead of actually looking at the data, he just looked at, well, the average person has it like this, must be right. I'm like, no, you have to look at the data. And there are lots and lots and lots of similar issues that just happened. I don't really want to go through all the bugs. And now the scary part is, the reason I found 7 bugs was that Claude gave me back the results, and I'm like, no, that's wrong. My lived experience with alcohol proves it, that your results are wrong. And then I just had it iterate, and oh, there's a bug, oh, there's a bug, oh, there's a bug. And now the scary part here is that I was the expert feedback loop for interpreting my own genome. But okay, that was for a fun alcohol. What happens if I try to have Claude interpret my genome for a disease, for something that's not obvious, that I don't feel? Then it just gives me back a result, and I'm like, okay, probably, possibly, probably not, I don't know. And that's really the scary part, is that the moment you lack the feedback loop, you're blind, and you have no idea what's happening. And the question is, where does this error come from? Can we somehow fix it? And you won't see anything in those slides. That's kind of deliberate. But whenever you want an AI, whenever you want to touch your own genome, you will operate at least on 4 files. You will have a diff that's about 1.5 gig in size. You will have the reference genome that's about 3.5 gigs in size. Some annotations just mapping what's inside the genome, what genes, where are they. That's, again, 1.6 gigs. And you will download a database of interesting markers that's like 200 plus gigabytes in size. And these are raw data. I mean, this is stable 100% data. LLMs are super, super good at finding structure in language. So whenever you just dump a lot of research papers to them, they will absolutely find all the structure, all the links, everything. But once you take away the language from them and you just leave the raw data, they're blind. I mean, they just no way to interpret that. Just a little parenthesis. Yes, maybe there will be some AI model that's not an LLM that will be able to number crunch on genetic data at some point. But LLMs which number crunch on linguistic data, it's not going to happen. And this kind of means that if you want to throw your LLM at that data, forget it. It's never going to interpret it. The only solution that we can do is we need to somehow make it deterministic. And that's, if you think about it, that's exactly what LLMs do when they program. So they generate some source code, but they never start generating machine code. So they write a high-level program, and then they run a linter. They run a compiler. They run whatever static analysis tools. These tools have a response, and then they iterate. In the case of trying to have LLMs actually touch your genome or do anything with your genome, essentially we need to reproduce these deterministic layers. LLMs are very, very good, kind of like this last mile technology, which just does the inference on the links, on the relationships. But everything always below that, we need to make it deterministic. And that doesn't mean that we can't have LLMs work on different layers. Yes, we can LLMs work on compression algorithms. We can have LLMs work on data set analysis, on interpretations, on finding relationships. You can have it work on everything, just not everything at the same time. So you cannot have an LLM cross-cut through the entire vertical. And essentially with the whole dark bio project, one of the things that I'm trying to do is to make it as deterministic as possible. So the LLMs can use the final mile. They can define the most interesting relationships, but we want to provide all the necessary deterministic tools for LLMs to just be able to access that data directly at a high level. So instead of telling the LLM that, hey, here's my data, read it and interpret it, what I want to tell the LLM is that if you are interested in that specific genetic marker, I can give it to you. If you're interested in some other genetic marker, I can give it to you. If you're interested in a gene, a region, something, I can give it to you. Then the LLM can actually operate meaningfully on that data. And the other problem that we highlighted here is that LLMs are kind of hallucinating slot machines. Now, I'm very, very bad for saying that. But essentially, they make mistakes. They make a lot of mistakes, and if you have an if you have a feedback loop, then those mistakes get gradually ironed out. Now, in the case of genomics, there's a problem. As I said, with the alcohol, you can be the expert because you feel how you react to alcohol, but once you start entering diseases, you're not an expert anymore. And the LLM is not an expert, and there's no deterministic process which will be the expert. And that's the other thing that we would like to address with the dark bio project is to try to somehow reintroduce expert feedback loops, meaning that apps, so let's say the platform can support a lot of mechanical indexing, searching, etc. But once you go into interpretation, the platform cannot interpret it automatically. But if you're a biologist, and whether you use LLMs or whether you use it manually, if you can write an app that just does some, I don't know, early onset Alzheimer analysis, and you write an app that has a result, then you, as your resident expert on Alzheimer, can actually validate that that is fine. And if you can publish that as an app, then all the other LLMs can all of a sudden consume your expert knowledge, and you don't have to guess whether that is correct or not correct. And so the idea with the whole dark bio project is to somehow try to create an app platform, kind of an open ecosystem, where yes, at the very, very top, you have an LLM that can combine stuff that is simply not done yet, but let's try to somehow build all the lower layers deterministically, and let's try to somehow have biologists be able to contribute to the whole thing so that they can actually build it up. And of course, the whole challenge doing this for us is to be able to do that in a very, very secure way so that the LLM itself, even if you use LLMs and go wild, you can never, ever lose your data. That you have hardware guarantees, that you have cryptographical guarantees, sandboxing guarantees, and a lot of other stuff. All the crypto and all the security stuff, I'm very happy to talk about. I can show it to you. I can demo it. Everything works. But for now, I wanted to actually highlight how you can get the LLMs to do your bidding without getting some really weird results out. Thank you very much. Yes?
It's more like a comment proposition, like you can operate. I totally agree that LLMs shouldn't be used for this reasoning, like heavy reasoning, because yeah, it's more like a glue, because it's like hallucinating and stuff. And there will interface to interact with it. An interface can be LLM, but yeah, reasoning would be in formal language. Have you thought about the formal language or not?
Okay, a very quick summary, because I won't be able to repeat the whole question and note. So the question I mentioned, I guess, was about can we somehow use can we introduce formal verifications to somehow dampen the effect of errors? Can we have experts who have the domain knowledge create the proofs so that then the LLM generated code we can just prove that it's correct? My answer would be, in theory, yes, in practice, no, because at some point somebody reached out that they sent my white paper to some biologist professor. They were curious what the opinion of the professor is. The professor skimmed through it and they're like, cute, infrastructure, we don't care. We want to do biology. And I think that's a thing that every single biologist will tell you is that, well, my friend, he's doing eye research. He really enjoys dissecting eyeballs. It's weird. He likes it. He won't care about creating proofs for proving his research that an eyeball works like this or that. So what he would want to do is just focus on doing his research. And I think what the ideal goal of of a platform like this is to basically go to these biologists and just speak their language and try to have them transplant their ideas as simply as possible into something workable. Now, if you can somehow automate transplanting their ideas into proofs, I think that's valuable. That would definitely work. I guess my note is that you cannot expect them to do it for you. I mean, they will absolutely stick to biology and not care about anything else. So I think that so my answer, I guess, would be that would be an ideal world if you can pull it off, if you can build the tools to give that to a biologist.
Files didn't work, but then the individual bugs were accounted for it could be identified. If this, like another solution, be structuring the context of all the files into something more queryable, so the LLM can do much more specific asks, and therefore we're not getting deterministic, but at least way better outputs.
Yes. So the question is whether or not the whole problem is simply that raw data doesn't fit into a context and it's just too much, too dense. And yes, that is the underlying problem with LLMs, that is just too much data. And that is actually the solution that we're also producing, is to create a lot of various granularity indexes, and then the LLM can just go and pull the exact data out. So what we want to prevent, I want to prevent the LLM from doing number crunching. Everything that does number crunching should be deterministic. I only want the LLM to do the last mile, just finding relationships. The asterisk there is that if the LLM does a mistake, how do you verify it? And that's where I think we do need to involve biologists, and we do need to make it very, very easy for them. So I don't think it's completely realistic to just rely on the result from an LLM, even though I would be happy to not have to actually double check or triple check something. But I think at least while we are in the world of LLMs, we will have these hallucination problems. And again, maybe there will be some other AI model. I know that there's isomorphic labs, which have their own research, their own AI, and that will probably be something completely different for LLMs. Maybe that will solve it, possibly, probably, I don't know. So it's a very, very promising field, but at least where we are currently and the trajectory we're on, I think we're not on the trajectory of solving it completely. So the question was, did we try putting some graph knowledge below the LLM, some to give it structure? And I haven't tried doing that. I think with LLMs, the reason you don't need that is because that, at least from my perspective, that's exactly what LLMs are super good at, that they can dig up the structure from research papers. If you make something a bit more structured, it probably helps, but I haven't tried it. So that, I think that would be a very, very interesting feat to try to somehow have something that's linguistic, but still more structured than just the free flow text. Okay, so I haven't tried it. I think it's a very interesting idea. Do we have time for? Okay, one more.
There's, let's say, your eyeball example, there's 1,000 research papers on genomics of eyeballs, and the work, part of the work to collect the data from the research papers really only has to be done once, or at least. If the elements get better, you might want to do it again, but are you channeling, let's say, these huge portions of research data into something that will be more digestible in the context?
I think the question is kind of similar with the context graph. The reason if you just do genomics, then probably yes, you could pre-process it in some way and then prepare something that's more easily applicable. But I think the really elegant thing of this whole model is that genomics is probabilistic. It kind of tells you that you are prone to some disease, you are prone to some issue, but it really also depends on your lifestyle choices. So yes, your genomes can say that, well, you are prone to elevated cholesterol level, but if you're athletic, then it doesn't matter because your body just clears it all out. And what this whole project can help is you don't need to stop at genomics. You can extend it to blood work. You can extend it to wearable data, various other stuff. And the power of LLMs is that they can actually look at what data sets you have available and then try to combine them. So they can say that, well, you have blood panels available beside your genome. Okay, instead of just looking at the genome research, let's also pull in the blood work research. Let's try to cross-reference it. Or if you have sleep data available, I don't know, maybe there's something valuable there. And I think if you try to approach it at the very top level from an AI perspective, they have this flexibility of just figuring or finding relationships. Whereas if you try to pre-process and structure it, you can go very, very far, but you can't really, you will never be able to create this query data set which is applicable to the same people. I mean, for everybody, because maybe I don't have blood panel. Maybe I use Whoop instead of Garmin. Maybe the data sets are a bit different, and then somehow combining them is not so deterministic, not so obvious. So I think the LLMs will always have this edge that they can just improvise.
So you don't want to pre-process signals before, because the LLM will find the signals?
I think it's more like it's a big can of worms, and then if you start doing it, then you're supposed to do it properly. And the LLMs also have the promise that they will do that fairly well. So the question is, where do you draw the line? How much can you just outsource to them? Even if they're not perfect now, maybe they will be better in a year. And so is it worth it your time, or is your time more worthwhile to create the tooling that the LLMs in 1 year can consume, or that LLMs in 1 year will still need to consume? But these are just completely open-ended questions. So I think the idea is that people need to try stuff and see what works and what doesn't, and I don't know, YOLO, iterate. Cool. Thank you very much.
Thank you, Peter.